Okay, good morning. Does anybody have any questions about the material from last time? Okay. So, to refresh in your memory, what you saw last time was that there is a lot of regulation going on of metabolism. So you learned about metabolism, that you have all these biochemical reactions in cells. And these reactions are mediated by enzymes, which are proteins. So we have all these biochemical reactions, and they may be going on at different rates, and this is all subject to regulation. So why is there regulation? Why are all these processes regulated? It's efficient. Why would you have a reaction if it's not needed at that time? But also, you don't want all these reactions going on at full force all the time and everywhere. That would just wreak havoc. You only want things to happen when they need to happen, and you only want them to happen at the rates at which they need to happen. Otherwise, you may get incompatible things going on at the same time. So that's why regulation is needed. And regulation is needed at the organ level, at the tissue level, but also in a time-dependent manner depending on the environmental conditions, whether there's light, whether there are certain ecological needs like attack or some symbiotic partner. So the plant constantly has to sense what's going on and then has to respond to it, and that happens by mediating the levels of different enzymes, but also the activity of different enzymes. So what you saw last time was enzyme abundance can be regulated, for instance, at the transcription level. There are transcription factors, which are proteins that lead to the transcription of suites of genes. And those suites of genes usually work together to make something happen. So you may have defense response genes or metal tolerance genes or photosynthesis genes, and they are commonly regulated by a common transcription factor. So when the need is sensed, that transcription factor is produced or activated, and then it starts turning on the genes that lead to an effective response. So enzyme abundance can be regulated in that manner via transcription. There can also be regulation at the level of mRNA stability or mRNA breakdown, so that is sort of a negative regulation. Sometimes messenger RNAs are actually actively broken down again. You can also have translation, regulation at the translation level, so to what extent is translation happening for a certain mRNA, and also at the protein stability level. Some proteins are actively broken down when they are not needed anymore. And then you also have regulation at the level of enzyme activity level. So you may have two versions of an enzyme. One is active, one is inactive. And usually, phosphorylation is a very common mechanism. And if an enzyme gets phosphorylated, that typically activates it. And when it gets dephosphorylated, it gets inactivated. So what that means is a phosphate group is added to one of the residues of the amino acids, and then it activates the enzyme, or it can be taken off again. So phosphorylation, dephosphorylation is a common mechanism of regulation. And there are, again, enzymes that do this. So enzymes that phosphorylate other molecules are called kinases, and enzymes that dephosphorylate them are called phosphatases. So often you have proteins regulating proteins, and so you have a whole cascade of proteins regulating each other. So it's complex, but the complexity gives it subtle ability to subtly respond to the different conditions. And so once you have an active enzyme, then again the enzyme activity is also determined by how much substrate there is for that enzyme. So there may just be, the enzyme activity may be limited by the amount of substrate there is, which is the metabolite that the enzyme works on, that it converts into something else. Or there may be the cofactor of the enzyme may also be limiting if there's not enough of a certain cofactor. Often enzymes need, for instance, metals or coenzymes. And then there are molecules, metabolites often, that can either inhibit an enzyme or that can stimulate an enzyme, and they're called inhibitors or activators. And they can work competitively or non-competitively, depending on where they bind on the enzyme. You have competitive inhibition if it binds in the same place as the substrate, the active site, and a non-competitive inhibitor binds in a different place on the enzyme. So this is all complex, but I think it's good to try to understand this, because once you understand it, you have a very broad overview of how metabolism works in general, not only in plants, but in any organism. So I just wanted to make these concepts clear. So enzymes are regulated in this manner, and often if you have a series of enzymes that work together, then we call this a pathway. You have pathways that produce something, so biosynthetic pathways, and you also have pathways that break down something, degradation pathways. So an example, photosynthesis could be considered a pathway, or respiration could be considered a pathway. Plants make all kinds of compounds. They're so chemically diverse, so there are all these different pathways that make these different compounds. They work together like an assembly line, because usually an enzyme can just do one little thing. It can just add one group to a molecule, to one particular molecule. So if you want to make a fairly complex molecule, you have to use a series of events. Like if you have an assembly line in a factory, you may have 5 people in a row, and one person does this, and another person does that, and so they each do one little thing, and then you get a final product. So going back to regulation, pathways are often regulated at what's called a key enzyme, which is often the first enzyme. And this enzyme controls the rate, the flux, through this pathway. And this enzyme is subject to, for instance, activation and inhibition, which is efficient. If you want to turn on and off a pathway, it's good to do that at the beginning of the pathway, not somewhere at the end, because then you would just have some intermediate accumulating. So you might as well do it at the beginning. So in this case, as an example, you have a pathway where we go from one amino acid, threonine, to another amino acid, isoleucine. There are 5 enzymes involved, and the end products inhibits the first enzyme, the key enzyme of this pathway. If you have a lot of the end product accumulating, then that enzyme, that first key enzyme, is inhibited. So that's feedback inhibition. Enzyme is inhibited by its product. So if the product accumulates, the enzyme gets turned down, which makes sense. Otherwise, it would just futilely keep making more of it when it's not needed. And vice versa, if there's less product because it's all used up by other processes, then we need more of that product. So then that feedback inhibition is alleviated, is turned off. So then the enzyme kicks in again. So that concludes this module 2, which was a recap of basic metabolism, enzymes, proteins in general. And you will learn more about different kinds of membrane proteins. I didn't say too much about them yet in module 6. So that is module 2. Any questions now about module 2? And just to be clear, modules 1 and 2, I did mention they were recaps. You probably heard about them before, but it doesn't mean they're not part of the exam material. They are part of the exam material. So now we're really starting to go into plant-specific processes, and we're going to look at plant nutrition in this first section. Plants have to feed themselves. They have to eat, they have to drink, like we do. So they take up water from the soil, and they take up inorganic nutrients from both the soil and the air. So we're going to have a look at how they do that, and some important regulatory mechanisms and also physiological mechanisms. How does it work? How do the plants move water around, for instance? And how do they move nutrients across membranes? So section 1 is going to look at the water and the inorganic nutrients that plants get from the soil. And then in section 2, we're going to have a look at carbon, which they get from the air, as CO2. Suppose that you don't know anything about what plants need, then the first thing that you could do is see what's in these plants, because that will give you a good clue to what they probably need, assuming that they have some control over what they take up. So if you look at what a plant consists of, it is 70 to 95% water, and then the rest is dry matter. So they need quite a bit of water. And if you would want to actually determine the percent dry matter and the percent water, what you could do is you take the plant, you weigh it, then you have the fresh weight of the plant, then you put it in a drying oven at about 70 degrees Celsius until the weight doesn't change anymore. Then you weigh it again, And then you have the dry weight. So now you know the dry weight and you know the fresh weight. And from the difference between the two, you know how much water there used to be in this plant. And that can tell you something about how much water it has, can tell you something about the water status of the plant, how happy is this plant. But also, if you really want to determine the growth of a plant, then usually you want to determine dry weight, because otherwise fresh weight can vary a little bit depending on if the plant was just watered or not. But if you have the dry weight, then the plant cannot cover that up. That's pretty clear-cut. So if you want to measure plant growth, you measure dry matter. So that's typically 5 to 30%. It depends on the plant species and on the plant organ. And then if you look further into this dry matter, then you see that it consists of a number of elements. And the most abundant elements are H, O, N, and C. So hydrogen, oxygen, nitrogen, and carbon. 96% of the plant consists of these elements. And among these, nitrogen is a distant fourth. There may only be 4% nitrogen or so, and H, O, and C are really the bulk. So that tells you that probably most of the molecules in the plant have H, O, and C in them. And that's true. If you think of, for instance, sugars, proteins, nucleic acids, fats, they all have H, O, and C. And then nucleic acids and proteins also have nitrogen in them. So the more proteins need a certain element, the more you're going to find that element in the plant. So if you look at who else is in this plant, the other 4%, most of the other 4% are 5 elements, P, K, S, Mg, and Ca. So that's phosphorus, potassium, sulfur, magnesium, and calcium. They're called the macronutrients. Macro means big, so they're nutrients that are needed in big amounts. So if you call those guys the macronutrients, you could consider H, O, N, and C the meganutrients. But nobody calls them that. So the macronutrients, and then there are trace elements, and there are 8 trace elements: iron, zinc, manganese, chlorine, boron, copper, nickel, and molybdenum. They are present in trace amounts. They're also called trace elements or micronutrients. And to give you an idea, the micronutrients are present at levels that are typically expressed as ppm, parts per million, so that's milligram per kilogram dry weight. And iron is typically most abundant. It's about 100 ppm, so 0.1, what is that, 0.01%. And molybdenum and nickel, they're just a few ppm. And the abundance, again, of each of these elements tells you something about how many molecules in the plant need this element. So molybdenum and nickel are present at really low levels because there are only very few proteins that actually use this element. The macronutrients are present in about a few thousand ppm usually. And then the H, O, N, and C is really percents or even tens of percents. So micronutrients, you see there are a lot of metals in there, and they are often cofactors for certain enzymes. So you can imagine that they're not needed in that large numbers. Whereas the macronutrients, if you consider, for instance, sulfur, that's present in most proteins. So you can imagine plants need more of that. Magnesium is present in chlorophyll, for instance, so plants need quite a bit of that. So how abundant it is in the plant reflects how much is needed. And that also tells you that the plant has control. The plant will take up as much as it needs, and then it stops taking it up. So it must have a sensing mechanism for each element, so it knows how much it has, and it knows if it needs to take up more or if it has enough. So the uptake mechanisms, the transporters for these elements, are subject to regulation. They can be turned on when the plant senses that it is getting low on a certain element. It ramps up its uptake for that element, and if it senses it has enough, it stops taking it up. So that goes by means of transporter proteins, which are again subject to regulation, similar to enzymes. So these are also called minerals or inorganic nutrients. Inorganic refers to the fact that they're not carbon-based. So, for instance, these elements typically are taken up in the form of ions like phosphate, sulfate, K+. So they're small ions positive or negatively charged ions, and they're not carbon-based forms, they're inorganic forms, these minerals. So plants feed themselves from inorganic nutrients, and then using those inorganic nutrients they can build up all their organic molecules, which, and by doing that they feed all the rest of life, because all other organisms depend on their capacity to feed themselves from inorganic nutrients, and then we can eat them, because we could never do that ourselves. But first we're going to look at water before we look at how they make these organic molecules out of inorganic ones. So water, it's needed as a source of these elements H and O, which are two of the most abundant elements, as you saw. They also need water as a solvent. Just all of biochemistry in cells is water-based. Everything is floating in water. So it's a solvent where biochemical reactions happen. And as you saw earlier when we looked at the vacuole, the water is also important for the structure of the plant. The turgidity of the plant depends on water filling up that vacuole, creating a positive turg or pressure. So for all these things, water status is important. If plants lose their water pressure, they cannot photosynthesize anymore, they cannot grow anymore. Eventually they will die. So, and maybe you know this, but water is the most limiting factor for plant growth on Earth, and this is both true in nature and in agriculture. So a lot of people in the world, a lot of researchers are studying plant drought mechanisms, plant water relations, because they try to breed plants that are better able to withstand water shortage and have a higher productivity. To give an idea, if we plot annual precipitation versus productivity, Fort Collins is somewhere here, and Western Europe would be somewhere here. And you don't reach this saturated level till about the tropics. So you can see how most areas in the world are limited by water. And this is a challenge also with the climate change in the Earth, because water distribution tends to get more irregular. It also shifts in different areas. Some areas in the world get more water, some get less water, but also the water falls more intermittently in larger amounts, so that creates periods of drought in between. So this is going to be a challenge that's only going to get bigger over time with climate change. Here on the left we see number of days with optimum water and corn yield. Again, you see a strong correlation. So we're going to have a look at plants and water, and we have two modules. In the first module, we look at basic concepts of cells and water. So what makes water move into and out of cells? And then in module 4, we're going to have a look at how this affects water relations of the entire plant. And then we're going to follow water from the soil all the way into the atmosphere. And we're going to see at all the different membrane steps and what makes the water move. But first, basic concepts of water and how this affects water movement into and out of cells. So water is actually a very interesting molecule. If you compare water with similar molecules like CO2 or H2S, then it is quite different, because it is liquid at ambient temperature, like room temperature, whereas most similar molecules are gases. And also water is polar, and what that means is that on one side of the molecule is a little bit negatively charged and the other side is a little bit positively charged. And this fact that water is polar makes it very good at dissolving molecules. And that also makes it very suitable to be a medium for life, because biochemical solutions in cells need to dissolve a lot of molecules, also very big molecules, which are hard to dissolve, like proteins. So it's a good solvent. Another thing that makes water very unusual is that ice, the solid form of water, floats on top of liquid water. And you almost never see that. Usually the solid form is more dense than the liquid form. But it's good that that happens, because if you have a water body like a pond or a lake, then you have a protective coat of ice on top, so life can keep going underneath. Water also has a very high resistance in temperature change. You have to put a lot of energy into it to increase the temperature, and also it stores a lot of energy, so it loses its temperature very slowly over time. Compared to other solvents, water has a really high resistance in temperature change. And this is also important because, again, if life happens in water and it's in a water body, then it's good if the temperature is pretty constant and doesn't fluctuate wildly. That's very hard for an organism to live in. So what makes all this happen? Water is polar. That gives it these properties that I just mentioned. And water forms hydrogen bonds. So what is a hydrogen bond? It's a bond between a hydrogen atom and another atom. The other atom is oxygen or nitrogen. So in the case of water, the hydrogen bonds are between hydrogen and oxygen. On average, a water molecule in a drop of water has about 4 hydrogen bonds with other water molecules, and these change all the time. So the water molecule is free to move around, and it just forms and breaks bonds, hydrogen bonds all the time. But because it has these hydrogen bonds, the water molecules tend to be fairly close together. They're kind of loosely, kind of like magnets, attracting each other all the time as they move around. And because of that, because of all these hydrogen bonds, water molecules in liquid water are closer together to each other than in solid water, in ice, because there they form these crystals, and where they are at fixed distances from each other, and those distances are bigger in ice than they are in water. So that's why ice floats on water. Because water is, let's first say, why is water polar? Oxygen has electrons that are unused and that are sitting on as it's shown here, on the top of this molecule. And electrons are negatively charged, as you know. So that creates a negative charge on that side of the molecule. Also, in the covalent bonds between oxygen and hydrogen, what a covalent bond is a shared electron pair between 2 neighboring atoms, where each contribute 1 electron, so now together they have a pair. But oxygen is kind of jealous. It doesn't really share very well. So it holds that electron pair close to itself and far away from the hydrogen. Hydrogen is not so good at also getting that electron pair. So that means that hydrogen is a little bit positive because it effectively has lost that electron a little bit. And oxygen is a little bit negative because it's holding that electron closer to itself. So water is one side is a little bit negative, other side a little bit positive, and that makes it polar. And that is why water molecules also tend to attract one another. In addition, that's what creates the hydrogen bonds also. This positively charged hydrogen from one water molecule is attracted to the negatively charged oxygen atom from another neighboring water molecule. So that kind of attracts each other electrostatically. So, and then the other reason why water is polar is because the hydrogens are arranged at an angle of 105 degrees. Whereas, for instance, in CO2, it's one carbon with 2 oxygens just in one row, 180 degrees. So, but because it's 105 degrees, this makes it such that the positive charge is really one side and the negative charge is the other side. So water is polar. Because water is polar, it forms hydrogen bonds. Because water forms hydrogen bonds, water molecules cling to each other, and they also cling to other polar surfaces. For instance, if you put a water droplet on a wall, you see that it kind of sticks to the wall and only slowly moves down. Or if you have a water molecule on a surface on a table, you often see that it kind of forms a flatter bottom. The top is round, but the bottom is flat because it kind of likes the table too. So it tends to cling to all polar surfaces. And the same is true for, in the plant, cell walls. The cell wall has polar groups as well. So water will also cling to cell wall material. And this is important when water moves up the plant because there you have xylem, which is cell walls left standing. And that's called adhesion. That water molecule clings to other molecules, other polar surfaces that are not water. But that helps with the moving up of water in plants. As water is sucked up by transpiration, The adhesion to the side of the xylem helps so that the water column doesn't collapse. So cohesion, adhesion, that's because water forms hydrogen bonds with themselves and with other polar molecules. The fact that they form these hydrogen bonds also makes water a really good solvent, because they form those hydrogen bonds also with proteins, for instance, and that helps keep these gigantic proteins in solution so that they don't precipitate. So it makes it a really good solvent, makes it float, it makes it mostly liquid at room temperature. That's also because they like each other so much. If they're in the liquid phase, they're all holding hands kind of, and they don't go so easily into the gas. They don't escape to the gas phase so easily because they have all these hydrogen bonds. They're holding each other down kind of. So that makes it more liquid compared to other similar molecules like H2S or CO2. And it also resists a change in temperature because they're all holding each other. If you have a group of people and you all link arms, for instance, and now I'm trying to move you guys, so I'm going to push and shake, but you're all holding arms, like demonstrators or picketers or something, sitting down action, they're all holding arms to each other, and then the police is trying to drag them away, but it doesn't work. So they're kind of like that if water molecules are all holding onto each other. So if you put a flame under a pan of water and it takes forever for it to boil, that's because if you want to increase the temperature, you want them to start shaking, right? Because motion is heat. Heat is molecular motion. You want them to get into motion by putting that flame under the water, but it happens very slowly because all the water molecules are kind of clinging onto each other. So you have to make the whole mass of water shake. So that's hard. But so that works the other way as well. Once you have water and it boiled, it takes forever for it to cool down as well. So it also holds onto, because they're all shaking together, and then it takes long for them to stop shaking because they're all doing it together. So water is a very special molecule. That's the bottom line. So water movement in plants tends to be a passive process. It does not require energy, no ATP. It's driven by concentration and pressure differences. So water tends to go from a high to a low water concentration by diffusion. But also water tends to go from high to low pressure. And both of these are passive processes. And if we consider short distance movement of water in plants across membranes, then both of these factors, concentration differences across the membrane as well as pressure differences across the membrane, affect the water movement across that membrane. And we have a term for that process that's called osmosis. So osmosis is the movement of water across a membrane. And in plants you also have more longer distance movement of water across longer distances, and there's no membranes involved. In that case, it's simple bulk flow. And in that case, it just goes from a higher to a lower pressure. And examples of this are water movement towards the root and water movement in the vasculature, so in the phloem and the xylem. In the xylem, there are no membranes because the cells are dead. And in the phloem, there are so many plasmodesmata that effectively they don't really form a barrier. The membranes don't form a barrier anymore. So those movements of water are simply due to pressure differences. And those pressure differences can go from positive pressure to less positive pressure. That's the case in the phloem. Water is pushed through. But in the xylem, it actually goes from negative pressure to less negative pressure. So there's suction going on at the shoot because there's transpiration. The water is leaving due to transpiration, and that sucks up the water from the roots. So this pressure difference can be positive pressure pushing water through vessels or negative pressure sucking water through vessels in plants. And if you have water movement toward the roots, it's also kind of a suction effect because the plant is taking up water and bringing it through the plant. So we're going to have a look at these processes. Bulk flow is getting more relevant in the next module where we're looking at the whole plant, but osmosis is our first topic of interest because we're focusing on cells now. So if we look at a cell and we look at membrane transport, osmosis, membrane transport of water, then there are two paths that water molecules can take. They can actually go through the membrane itself in between the phospholipid molecules. So that is simple diffusion. Even though the membrane is hydrophobic, so it doesn't like water, water can still sneak through because it's small enough and it's not charged. But it goes really slow. So it would not be fast enough to satisfy the need of the plant. And that is why the plant also has facilitated diffusion of water. It has these channels, water channels, which are called aquaporins. So aqua is water, porin is pore, so it's a water pore. And it allows water to flow through the membrane from a high to a low water concentration. And in addition, it's not only concentration, but it's also pressure that affects the flow of this water. So osmosis is the passive movement of water across a membrane through the combined effect of concentration difference of water and pressure difference on both sides of the membrane. Clear so far? Water movement across the membrane osmosis is an example of diffusion, and just a few principles of diffusion, which you probably heard before, but it's good to reiterate them. It's passive movement of molecules, and it's simply due to molecular motion. So as long as the temperature is not absolute zero, which it never is on Earth, there is molecular motion. All molecules are in motion. So if you consider an unequal distribution of a certain molecule over a space, then simply due to chance, the molecules have a higher probability of moving from the area of the high concentration to the area of the low concentration. So if you consider here these green molecules, at the beginning of this experiment, the concentration of the green molecules was high on the left side, low on the right side. If you wait a little bit, then you see that there has been diffusion from the left to the right. At this point, it's still higher on the left than the right. So now all molecules continue to move, but still the probability is higher that they go from left to right than from right to left. So you see that represented by these red arrows. And if you wait long enough, now the concentration is equal across the space. And you see that the molecular motion still keeps going, and they're still going from left to right, but they're going at equal rates from right to left. So we have equilibrium. So when there's equilibrium, there's no more net diffusion. So there's still movement, but that doesn't change anything. They're equally going left to right and right to left. So when we have an unequal distribution of concentrations, so the concentration, if we're talking about solutions here, where the dissolved molecule is the solute and the water here is the solvent, so it's the liquid, we have a solution. The solution has a certain concentration, so that is number of particles per volume of solvent. If that concentration is different across a space, left-right, then there will be diffusion until there's equilibrium and the concentrations are the same across the entire space. So that's the concept of diffusion. It goes from high to low concentration until there's equilibrium. At that point, there's still movement, but no more net movement. And another thing that is good to realize is that if you have different solutes in one and the same solution, represented here by purple and green balls, then they diffuse independently from one another. So at equilibrium, we have equal number of purple molecules on the left and the right, and also equal number of green molecules on the left and the right. So diffusion of different compounds is independent from each other. And in this case, we see a membrane here with holes in it, and those holes are big enough for those particles to move through. And that's going to be different from the next examples that I'm going to show you, which shows also a membrane, but there the membrane is selectively permeable, where only the water molecules can move through, but not the solutes. The holes are too small for the solutes to move. And that creates an interesting situation. So osmosis is diffusion of water across a membrane, and water goes from a high to a low water concentration, and water goes from a high to low pressure. In this case, there is no pressure yet, but we will bring in the pressure in one of the next slides. But the first thing that we're going to change now is that our membrane here is semi-permeable. And that's also called selectively permeable when we're talking about biological membranes, because there the cell can regulate which molecules are allowed to go through and which are not. But in this case, we're looking at an osmometer, just a simple U-shaped glass tube. And in the middle, we have here just a membrane with holes in it. And the holes are too small for the solutes to move through, but the water molecules can move. We have here two solutions on both sides of this membrane. One has a higher solute concentration than the other. And there's a term for that, where the concentration is higher than another one. We call it hypertonic. And if a solution has a lower concentration relative to another one, we call it hypotonic. Hypo means under and hyper means above. So these are relative terms. You can compare them with tall. So I may be taller than one person, but I'm now not as tall as another person. It's all relative. Hypotonic, hypertonic. And then there's a third term called isotonic, and that is the term for when two solutions are actually the same concentration, and they're isotonic. Maybe you know that term from sports drinks, which are called isotonic relative to your body. So solute, solvent, those are also some terms that you're going to hear again and again. So solute is the particle that is dissolved. Solute is the liquid in which it is dissolved. So when you have two solutions, then you can express the concentration of the solute in relative to the solvent as you can express that as number of solute particles per volume per liter of solvents. And if that solute concentration is higher, then we say it's hypertonic. If it's lower, it's hypotonic relative to each other. If it's the same, it's isotonic. So now let's look at what's going to happen. If we start here on the left, we have two solutions with a different concentration with a semi-permeable membrane. The water molecules can move, solute particles cannot move. So what's going to happen? Different compounds diffuse independently. So the solutes would like to move, you could say, but they can't. How about the water? The water, wherever there are solutes, there's no water. So if you consider it from the point of view of the water concentration is higher where the solute concentration is lower, and vice versa. Okay? So on the left, the solute concentration is lower, so the water concentration is higher. So the water will diffuse from high water concentration to low water concentration, so from left to right. So over time, then this is going to happen. You see now the water has moved. The solutes have not moved because they cannot. The holes in the membranes are too small. If you would count the purple balls, you see the purple balls have not moved, but the water has moved, because water has gone from high to low concentration. At some point, there is equilibrium, because now the concentration is the same on both sides. So you can reach equilibrium by just moving the water molecules. And this happens in plants too, in cells in general. So water will always tend to go where you have the highest solute concentration. But what you also see here is that on the right side of this tube, you see a pressure buildup. Gravity is going to pull down on that increasing water column. That's going to create a pressure on this membrane. That's probably going to push back. So at equilibrium, the actual concentration on both sides may not be exactly the same, because pressure is going to push back. And you have the same in cells. In cells, you have water spontaneously moving to where the solute concentration is highest, but you may get a pressure buildup, and that's going to push back. And that's what you have in cells, in plant cells particularly. Because plant cells are under positive pressure. They have the cell wall that pushes back. So it's kind of like a tug of war. And osmosis is the outcome of this tug of war, of water wanting to go where the solutes are, but pressure often pushing back. That's kind of the bottom line. So water is moving across membranes toward the highest solute concentration. And I put here total with an underline under it. What you can do is you can just add up all the dissolved particles, and they all count equally well. So the more solutes you have, and it doesn't matter if it's a tiny molecule or a gigantic molecule. One particle is a particle. But it has to be dissolved. So if it's not dissolved, it doesn't count. But all the dissolved particles count as a solute particle, and they all attract water. And water will go to where you have the highest solute concentration. It will go from hypotonic to hypertonic. if you look at the two solutions on either side of a certain membrane, one may be hypo, one may be hypertonic, and then it goes through that membrane toward the hypertonic side. And plants can use this if they want to take up water from the soil. Then they want to try to be hypertonic relative to the soil. So they can concentrate a lot of solutes, like sugars, in their roots, and that will attract water from the soil. And it's represented here, if you consider this red line as a semi-permeable membrane, and these stars here, these asterisks, as solutes, then you see there are two of these solutes on the left, four on the right, so water will move to the right, to where you have a higher solute concentration. Okay? It's the number of particles that counts, and that's why when you talk about concentration, you talk about molarity, and a molar is 1 mole per liter, and a mole is 6 times 10 to the 23rd molecules, Avogadro's number. The good thing about mole, if you talk about molar, is that you can simply add up all these concentrations from all these different solutes. You may have like 0.1 molar of glucose and 0.2 molar of sucrose, where sucrose is twice as heavy, but it doesn't matter. You're just counting them as number of particles. And then you can just add up mole to mole. So you can just say, okay, so 0.1 plus 0.2, there's 0.3 total molar of concentration of solutes. And if you do that, then you can compare those molarities, those total solute molarities, and then you will see, aha, this side is the hypertonic side. It has the highest molarity. That's where the water is going to go. So considering another example, here we have 0.4 molar of glucose on one side of the membrane, 0.1 molar on the other side. So the water will go from the 0.1 to the 0.4. Where you have more solutes, you have less water, that's where the water is going to go. Now we have actually two different solutes, 0.1 molar glucose, 0.4 molar fructose, and on the other side, 0.4 molar glucose. These sugars can all not move through this membrane, so only the water can move, and the water will move to the left because we have 0.5 total molar of solutes, and that's higher than 0.4. So the more solutes you have, the more it's going to attract water via osmosis. And here we have, now this is an interesting kind of trick question. What if you have 0.1 molar glucose on one side and 1 molar glucose on one side and 1 molar NaCl on the other side? Then you might think, oh, 1 molar, 1 molar. There's going to be no net diffusion of water. It's the same. Not true, because salts dissociate. So if you have 1 molar of NaCl, it's going to fall apart when you throw it in water, and then you get actually 2 molar particles, or 2 mole per liter particles, because you get Na plus and Cl minus. Or if you would have magnesium chloride, which people put on their driveways when it gets cold to lower the freezing point, then you get magnesium 2 plus, Cl minus, and another Cl minus, because it's MgCl2. So you get 3 molar of particles for 1 molar of magnesium chloride. That's why it's good at lowering the freezing temperature, freezing point. So keep in mind, salts fall apart.
Changes the game?
What game? Salt changes the game?
Well, no. Are you saying that you have different counts?
I'm saying be aware that if you're comparing molarities, salts dissociate. They fall apart into particles, and they give more particles than is suggested by just the molarity of the salt formula. So that's one thing to keep in mind. So what's going to happen here? You actually end up with twice the number of particles per volume on the NaCl side. Water is going to move from glucose side to the NaCl side. Another thing to be aware of, and I mentioned this earlier, if things are not actually dissolved, they don't count. Example in the plant world, starch. Starch is not soluble. So there's lots of starch in plants, but it doesn't contribute to what's called the osmotic potential, which is the potential of solutes to attract water. So starch, not soluble, does not attract water. So if you think of implications for the plant, if a plant wants to store lots and lots of storage carbohydrate, you could say, well, I can see why it could be good to store them as starch. Otherwise, you may mess up the osmotic balance in the plant. If you would store so many molecules in this chloroplast, that may actually screw up. It may suck in all this water from the cytosol. It may screw up metabolism. So maybe that would make sense. But if you also think of if a plant has a choice, it's going to store starch or it's going to store, there are also fructans, for instance, which are soluble, or if it's going to store sucrose like sugar beet, or onions have a lot of glucose. So different plant species store different sugars, and some are soluble and some are not. And that's going to affect the solute concentration in this organ, and that's going to affect the plant-water relations. So if you consider an onion, that often has to survive a cold winter, and then it may actually be good to have a very high solute concentration. It may help protect the plant, this organ from freezing, from drying out, because the more solutes, the less it freezes, and the better it's able to hold its water. And if a plant is undergoing drought stress, or frost, or salt, which are all osmotic stresses, then often what you will see is that it turns starch into sugar. So it lowers its osmotic potential. It lowers it, meaning, I'll get back to that later, but it increases its solute concentration, and that enables it to hold on to its water and to attract new water from its environment. So a plant, by manipulating its solute concentration, can respond to changing environments like drying out. And you can sometimes even taste this. If you have a vegetable that has seen frost, it often tastes a little sweeter because it has made some sugars to withstand this osmotic stress that is frost. We'll see more about that when we're in the stress physiology chapter. Yeah? Yeah, so the question is, for starch to be converted to other forms of sugars, it would have to undergo enzymatic conversion. That's true. So this choice of what storage carbohydrate am I going to store, and when am I going to remobilize this, what will I turn it into next, those are all decisions that the cell can make by means of activating different enzymes. And you will see how that's regulated in section 2. We're going to see that example. So osmosis has implications for cells. So suppose that we have a cell, this is an example, that has a total solute concentration that's equivalent to 0.3 molar. Then if you throw that cell in a 1 molar solution, that cell is going to lose water, because the solution is going to be hypertonic. It's going to attract water from that cell. The cell is going to shrink and die, plasmolysis. If you throw that same cell in a solution that's 0.3 molar, that's isotonic, there will be no net diffusion. So water is going into and out of the cell at the same rate. The cell is going to remain its same volume. And if you throw that same cell in a 0 molar solution, pure water, that solution is hypotonic relative to the cell. Water is going to move into the cell. The cell is going to swell up. So how does the cell feel about that? If the cell is an animal cell, it's very sensitive because it doesn't have any protection. So it cannot withstand losing water. It will die. It cannot withstand gaining water. It will burst and die. It needs to be in an isotonic environment. So our cells have to be in an isotonic environment all the time. Our cells are bathing in fluid, tissue fluids, and that's maintained at a very critical isotonic concentration solution. Plant cells, on the other hand, they have the cell wall. They actually love to be in a hypotonic environment because then water will keep wanting to move in, and that keeps the cell nice and turgid. So plants want to be in a hypotonic environment. If you forget to water your plants, then they may experience this situation. They lose water until they are wilted, and that it can just concentrate the environment of the cell until the cell is wilted. And that doesn't kill the cell, but if the plant is wilted, it will eventually also die because it cannot keep its photosynthesis going, it cannot keep the stomata going. So this is, temporarily it's okay, but it shouldn't last too long. And if you really put the cell in a very high salt solution like seawater, most plants are going to croak because then all their cells are going to shrink up and die. So cells, plants typically like to be in isotonic environments. Their cell wall protects them from bursting. Okay, that's it for today.